Yes. We will now hear from Dr. Michael Monn. Dr. Michael has done his bachelor's, master's, and PhD from Brown University in Rhode Island. For his PhD, he worked with Professor Hanish Kesari on mineralized biological composites. He is currently with St. Gobain, North America. He works in their R&D division on aerospace composites. After Dr. Michael, we will hear from Dr. Zhen Yin from McGill University on bio-inspired architecture materials. Dr. Michael, the screen is yours. I will stop sharing my screen.
Awesome. Thank you very much. Everyone see me? Yeah?
Yes, we can see you.
Awesome. Wonderful, wonderful. Well, it's a real pleasure to an honor to be invited to partake in this this type of meeting to celebrate Professor Simha. I've never met him in person, but we had a very short interaction when we were practicing the logistics of this presentation. And a lot of what you just shared in your talk resonated really, really deeply with me. I think that there's I see the richness of the community that you've built, and it's really awesome to see so many people, you know, having you as a mentor through their careers in academia and elsewhere. I'm gonna share my presentation, and we'll see. Can everyone see my presentation then?
Yes, we can see your screen.
Okay, great, awesome. So yeah, I'm going to be talking about the work that I did some of the work I did during my PhD. It's focusing on a paper or some results from a paper I just published this year, actually. So the talk, as you see, is titled Unraveling the Connection Between Architecture and Toughness in Layered Biological Ceramic Composites. So it's a mouthful, but I hope we can break it down and really learn something new about biology, about the world around us, using fracture mechanics to make that kind of exploration. So I did this work in collaboration with a number of people, and I think that there's a lot of connectivity with what Professor Simha said during his talk. So my collaborators are Kaushik Vijayakumar, Sayaka Kochiyama, Jared Ferreira, and Zhenjie Yang. Kaushik actually did his master's at IISc under Professor Narasimhan, and he was my batchmate in PhD. We worked with Professor Hanish Kesari, like Ram had mentioned, and you know, I think that what you said, Professor Simha, about mentorship and what you've really demonstrated through your mentorship has resonated with me through the type of mentorship that I had received from Hanish, and that still really sticks with me today. The other part of your talk that really resonated with me is your discussion of the ocean and how little we may have dove into its depths. And so while I thought that was a great metaphor for the career trajectory and path as a researcher that you were explaining, it has a lot more literal implications for what I'll be talking about today. So I'll start off just really talking generally about biological materials. So layers are really a prevalent motif in these biological ceramic composites. So what I mean by a biological ceramic composite is something like a seashell. So if you look at a seashell, like in figure 8 here, this is a red abalone, and you were to cross-section this iridescent layer called nacre and look at it under a scanning electron microscope, you'd see something like figure B, where you have this brick and mortar structure. The bricks are composed of a calcium carbonate mineral, and in between the bricks, there's a really, really thin layer of a softer proteinaceous material. This motif appears in other types of shells from other families, taxonomic classes, and like this conch shell. And in this conch shell, the layered structure is a little bit more complicated. It has some hierarchy. There are layers within layers, and within within each one of those hierarchical levels, you have this recurring sort of motif of ceramic bricks surrounded by very, very thin protein layers. We even see this in our own bones. So if you were to etch a cross-section of bone, you would etch away some of the mineral, the hydroxyapatite, and you'd see that we also have this alternating structure of mineral and soft proteinaceous material. So what's very interesting about this architecture is it leads to very drastic changes in mechanical behavior. So this shows some tensile tests. This is from Professor Bartholat, who the next presenter was one of his students, I believe. These are the tensile tests performed on pure aragonite, which is that calcium carbonate mineral that makes up those tablets, as well as nacre, both dried and hydrated. And while there can be a huge discussion about the role of hydration in these materials, I really just want to focus on the hydrated case, because that's the native case, how the shell actually performs in its native environment. And you can see that the mechanical response of these materials is very, very different. The aragonite is typical of a ceramic. It's fairly linear in stress and strain up until catastrophic failure. Whereas the nacre has this almost elastoplastic response. And what's really amazing about this is that this response manifests despite the fact that the ceramic makes up over 98% of the material by volume. So a very, very small amount of this soft proteinaceous material, when added in the correct way with the correct geometry, can lead to this extraordinary mechanical behavior. What this leads to is a huge amount of energy dissipated in these biological materials. So here I'm plotting, you'll see this average R, and we'll get into a bit of that later. You can think of it like work of fracture. So this is the energy dissipated to cleave the material into two pieces. And that can sit for many biological materials in the range of 1,000 to 10,000 joules per meter squared. I think if I try to, oh, I can't even see my cursor. Yeah, that's fine. And what's really wild is if you were to compare this to the ceramic constituents that make up these materials, the calcium carbonate, the hydroxyapatite for bone and antler, the enhancement in fracture toughness is truly remarkable. This is often several orders of magnitude and has really inspired people to try to understand the mechanisms through which this architecture enhances toughness, with the hopes that we can apply these same concepts in engineering context with engineering materials. So I actually did, I studied primarily a sponge called Euplectula aspergillum, and I use this as a model system for looking deeper into structure function connections in layered biological composites. So this comes back to what Professor Simha was saying about the ocean. These sponges live deeper than we've really been able to explore in the flesh. They live on the ocean floor at depths of about 1 to 2 kilometers. And they feed by filtering microorganisms out from the seawater. So they pump water through their bodies, and they use specialized cells to filter out small microorganisms which they eat. These sponges, particularly Euplectula, has a very intricate mineralized endoskeleton, much like our own. We have bones, they have their own version of bones. And this skeleton is a very complex assembly of glass fibers that we call spicules. So this is a picture of one of these Euplectula sponges with all of the flesh, all of the tissue removed. So in figure A, you see the mineralized endoskeleton. So that skeleton is glass, or roughly 99% glass by volume. And at the bottom of the sponge on the left-hand side here, that's the part that would anchor in the mud on the seafloor. You see these hairs, these hair-like fibers. Those are also glass fibers, and they act like roots. They're called the basalia spicules, or the anchor spicules, and they serve to mechanically anchor the sponge to the substrate of the seafloor so that it can pump water through its body and survive. What's very interesting is if you look at one of these spicules, they're about 50 microns in diameter, about half the width of a human hair. And if you cross-section them and you look at them in a scanning electron microscope, you can see that they have a very intricate layered architecture. This architecture is different geometrically than the architectures that we saw before in nacre and bone, but it has the same type of motif, which is alternating layers of a ceramic component and a proteinaceous component. And the volume fraction of the ceramic component is very, very high. So for the spicules, the ceramic component's about 99% by volume, and the proteinaceous layers in between these ceramic layers are on the range of 5 to 10 nanometers thick. So taking a closer look at this tree ring-like architecture, we can see that it consists of a central core that's about 20 microns in diameter, and then a number of silica cylinders. While they look fragmented, like they might be sections of a cylinder, that's just due to the process that we have to saw the spicules in order to see this cross-section. The cylinders are actually intact, and there are about 25 of them in each spicule. And you can see if you take even a deeper look, this was some work by James Weaver at Harvard, which I collaborated with a couple of years ago on a paper. He looked specifically at this proteinaceous layer, which you can see in subfigure B, a fibril that's spanning two separated silica cylinders. So early on, what interested us very much in this structure, in this animal, is that if you look at a number of spicules from a number of different individuals from this sponge, so this is showing 4 different individuals, 4 skeletons, and a number of spicules, n equals 35, 23, 21, 37 from each skeleton. We measured the inner radius, that is the radius of the core, the outer radius, so the diameter of the spicule, or the radius of the outside of the spicule, and the number of layers in the spicule. And you can see, looking at this, this table, that if there are any people who are biologically inclined here, this is a very remarkably consistent. That this is not something that varies across different individuals, that is driven predominantly by some external environmental factors. This architecture and the sizes and the scaling of the architecture is really programmed by the animal. So this consistency really drove us to ask the question, does the architecture provide a mechanical benefit to the sponge? And if so, what is that benefit? So previous researchers have suggested that these layers are enhancing the spicule's toughness, fracture toughness, like we observed in nacre. This was based on the fact that the architecture was visually similar to that of nacre. And also when you did 3-point bending tests on some related species, so this is, if you look in subfigure B, the gray curve is from a different species of sponge that also has layers, albeit much more of them, about 500 layers in this case, that they have this jagged force displacement response, which they attributed to a progressive failure of the layers and an increase in energy dissipation, and that the layers were providing some mechanical benefit to the animal by enhancing the energy dissipated during failure. This has never been investigated for euplectula spicules. This has really been attributed to them. Several researchers have made claims that the spicule's layered structure enhances their toughness, but there's never been an experimental investigation of that enhancement. So that's what we set out to do. And the first step in that process was constructing a mechanical testing device that could measure the flexural behavior, flexural response of these spicules, and measure the energy dissipated during the propagation of a crack. So to do that, we needed to measure forces in the range of micronewtons to millinewtons, and displacements on the range of microns to tens of microns. So we didn't find an instron to be sufficient. Well, all of you are muted. I hope several are laughing. And also something much smaller like an AFM couldn't provide the maximum forces that we needed in order to do this. So we basically designed a scaled up AFM. So this scaled up AFM has a sample stage that we place our specimen on, and the specimen spans a trench in the stage. And then we come down from above with a wedge, a tip, just like an AFM tip, except quite a bit coarser. That tip is attached to a cantilever that we measure the stiffness of, and the deflection of the cantilever is measured with a fiber optic displacement sensor. So this is basically a homebrew scaled up AFM for doing these flexural tests on micron to tens of micron diameter fibers. So when we perform our bending test, we move the stage up and we measure displacement of the stage W sub S. In the process, the cantilever deflects an amount W sub C, and we measure the force based on the cantilever's stiffness and its deflection. And we can compute the deflection of the load line, or the displacement of the cross-section of the spicule W naught based on those measurements WS and WC. So you'll see the configuration that I've shown here is a little bit different from a standard 3-point bending test. You have the specimen shown in blue, the stage shown in gray, and the edges of the specimen over the stage have this green sort of shade to them. And that's glue that we have applied to the edges of the specimen. So we've glued the specimen down so that it isn't a 3-point bend test, it's kind of a double encastered 3-point bend test. And this serves two purposes for us, which one of which I'll discuss a little bit later, but one is very practical. These fibers are very, very small, several millimeters long in the sections that we've cut, and about 50 microns in diameter. So handling them can be very frustrating, and gluing them down helped a lot with that. So this stage was moved in 1 micron increments, and we measured the cantilever deflection, as I said, with a fiber optic displacement sensor. And then we computed the force and the specimen deflection W naught. So that shows our measured quantities and our computed quantities. So to discuss a little bit more about the specimen, our gauge length is about 800 microns. And if you look, you can see there's a notch cut in the specimen right under the wedge that we apply a load with. And we cut that notch with a gallium ion beam. So you can see sort of two dotted lines here, a green one and an orange one, and we cut it in 2 steps. That's because we were trying to cut rather deep notches. And when you're cutting with a focused gallium ion beam, the gallium ions ablate material, and that material can sometimes redeposit. And we didn't want that, so we started with a sort of wide coarse notch and then created a very sharp notch at the tip. So I've been showing you schematics this whole time, but we should look at what it actually looks like in a SEM. So this is sort of the picture of a representative sample that we had. And you can see if we zoom in, that 2-stage notch with a total length A. The notch root radius at that tip typically was on the order of 50 to 100 nanometers. Which is smaller than the smallest microstructural length scale of the spicule. So here's, again, to start getting really into the experiment, a video of the experiment on the left. You'll see this horizontal line, that's the spicule. It's illuminated collinearly on axis, so that's some reflected light that you see off of the curved surface. And then the right-hand side, you'll see a force displacement curve following the video of the sample deflecting. This is sped up 48 times, and you can see this is the response that we get. So we performed a load and then an unload on the sample. I'll show you that one more time. A load, and then we unload the sample again in 1 micron increments. If we take a closer look at the force displacement curve, this is where we start to see some very interesting features. So you can see initially the curve increases, and then there's a small drop. This is a pop-in event associated with the initiation of crack growth from the notch root. After that crack growth initiation, the response becomes nonlinear until we reach this green square, which I'll mark as a complete failure. And I'll describe why we think that is the point of complete failure. For a particular specimen, after the crack has grown all the way through the cross-section and completely cleaved the sample in 2, there isn't a total loss of stiffness. You actually end up with a configuration that looks like this. So you have 2 cantilevers that are being displaced. So that should lead, at least initially, for small deflections, to a linear force displacement response, which is what you see after that point of complete failure. So that nonlinear region between the crack growth initiation and the complete failure, we believe corresponds to the region in which the crack is growing across the spicule's cross-section. And once you reach complete failure, which is really the point of minimum elastic compliance, that's what you would expect as the crack grows through the cross-section, the compliance of your specimen will decrease monotonically until the crack completely cleaves the specimen. That's how we define our complete failure point. And you can see that it lines up pretty well with the unloading, which is those light gray points. There's a small residual displacement at the very end of the unloading, and that's due to the fact that as you unload, the two halves of the spicule don't completely intermesh again, and there can be some interaction between the two tips of the cantilevers that prevent them from returning to their the same place as the reference configuration before we started the experiment. So from this, how are we going to quantify toughness? How do we measure toughness? I really looked at this from the perspective of a crack growth resistance curve. So a representative crack growth resistance curve with a rising R response is shown here in pink. And that shows that as the increment of crack length, delta A, increases, the crack growth resistance also increases due to a number of factors. We're not trying to necessarily discover the mechanism here, that'll come a bit later, but really capture the effects of the mechanism on the energy dissipation in the system. This increase in R can be due to a number of extrinsic or intrinsic factors that occur both behind or in front of the crack tip. So rather than trying to measure the entire R curve, which can be complicated with materials that have extrinsic toughening mechanisms, we're going to characterize it by two specific points. That is R at 0, which is the fracture initiation toughness by another name, and by the average value. The fracture initiation toughness should be a material parameter. It should be a constant, or roughly so. Not necessarily the case in heterogeneous materials. And the average value of R is not. This corresponds to work of fracture. It can be shown that average R is actually twice the work of fracture. And that can be size dependent on the material. It can be sample geometry and loading configuration dependent. So this we need to be careful when we're making comparisons with. So I'll start with how we measured the average crack growth resistance. As I said, average R is twice the work of fracture, which is the total energy dissipated in that fracture process divided by the cross-sectional area of the ligament. And so we can compute the work of fracture using an incremental energy balance, where on the left-hand side of this equation, you have the total extrinsic work done on your system. On the right-hand side, you have some elastic energy, that's U sub EF, and your energy dissipated by fracture, so UF. In many cases, the elastic energy is going to be 0. For example, for a standard CT test or a standard 3-point bend test. Once the sample has failed, it is no longer carrying any sort of load, so your elastic energy will be 0. In our case, that's not the case because of the cantilevers that I showed you previously from when the sample is in its completely failed state. So the left-hand side, the external work done, can be found by the area in between the loading and unloading curves, or more cleanly in this case, the area between the loading curve and that line of minimum elastic compliance drawn to that green dot, that point of failure. And the elastic energy, I can't see that equation, it's kind of hidden by some views here. Maybe I can minimize this, no, okay. Anyway, I think it says, force times, sorry, I keep flipping through. There we go. Yeah, so force times the displacement divided by 2. So we use this to measure the average value of R, and you can see that it's in the range of, there's some scatter, and it's in the range of roughly tens to hundreds of joules per meter squared. And here it's plotted as a function of the notch length A by the spicule diameter D. We don't see a really clear trend in A by D. So this is for our spicules. And this is what you'd see out of a human-made glass, where average R would typically be around 10 joules per meter squared. For the initiation toughness, we go back to the energy release rate, which is the derivative of the potential energy of the system by an increment in crack area, delta big A. And this depends on the shape of the cross-section. So if we recast this as a derivative of potential energy with respect to a crack length, you also have this second term on the right-hand side, delta big A, d delta small a, which captures the cross-section, which is a little complicated to use the circle rather than a rectangular cross-section. So Griffith's necessary condition for crack growth says that crack growth can occur when your energy release rate exceeds your crack growth resistance. And using that and taking delta A going to 0, we can write this equation as your initiation toughness, R0, will look something like this, where the first term captures that geometry effect, and the second term is that change in potential energy. So we get that second term from from a computational mechanics simulation. In that simulation, we apply a displacement W sim, and we can do this rescaling with the WC by W sim out front here. So in our experiment, we measure the deflection of our cantilever at the initiation point. From our simulation, we use a 4-point central difference to get a change in potential energy with respect to a crack increment. And we measure these geometric quantities from the micrographs. So here we can see the initiation toughness, again, as a function of A by D. And this increases as your A by D becomes very, very small, less than 0.1. But in a middle range of A by D from, let's say, 0.2 up to 0.7, it's relatively constant with a value between 1 and 10 joules per meter squared. Again, human-made glass is shown to have initiation toughness around 7 joules per meter squared. So how do we quantify?
Sorry to interrupt.
Yes.
Yes. We are close to the time. I have a couple of suggestions. We can continue during the break. I am hooked. Okay. We can.
Okay.
If you would like to hang around after about half an hour, we have a 30-minute break. We can continue the presentation, and I want to listen to it. I mean, I can hear Hanish coming through your voice. Right? So that's one option. I mean, if for the benefit of for the audience who will leave for the break, if you want to just show a concluding slide, we can have that now. But I mean, I hope that you will stick around during the break and give us the rest of the story.
Yes, of course. So I will show you a concluding slide then, and it spoils it a bit, but that's totally understandable. And yeah, I'll hang around during the break as well if anyone's interested in hearing the last couple slides.
I am very interested in 100%. I mean, we've been...
Okay. So what we really found is that compared to these other materials with layered architectures, like nacre, bone, antler, and conch shell, the enhancement in fracture toughness, both in initiation toughness and in this average R sort of metric for toughness, is very, very small. It doesn't really compare at the same scale. You don't see the huge enhancements that you saw in these other materials. An enhancement of, let's say, 2 to 10, a factor of 2 to 10 is still very impressive in an engineering sense. But it really leaves this question mark, this mystery as to why this particular material doesn't perform like these other materials. The secret to that lies in the geometry of the layers, the curvature of the layers. So I guess I'll end there, and if people want to hear more during the break, I will stick around.
Yes, please, certainly. I mean, I would request the audience to stay back in half an hour's time so that we can listen to Max connect the dots, right? from where he left off, right? Thank you, Max. I mean, I hope that, I'm going to be here. I hope that you will also hang around. Max, if you're still there, and if you'd like to complete your talk, I'm here to listen.
I am. Thanks, Rob. if anyone is still interested, there were, I think, only about 3 slides that I didn't show, but I received a couple questions in the chat, and I think it might answer a few of those questions. So I'd be happy to share. Up there. Okay. Yeah. So I stopped here, which is really where I was
yes,
sort of getting to the idea that we need some sort of a metric to compare this material to. Because at this point, I've been showing these lines for human-made glass, but really this isn't just standard glass. It's chemically fairly different. It's partially condensed silica. So that means that it is not just SiO2 fused silica. It also has some end groups, like some hydroxyl groups. You can see that in this single pulse silica and NMR spectra. So the euplectula spicules I've been looking at is shown here. And these are spicules from other sponges that are related to euplectula. Some of them have layers, but one that doesn't are these spicules from a sponge called Tethya aurantia, and that's shown in figure A, a cross section. So you can see it's monolithic, but it is chemically basically the same. These curves are just vertically shifted to show the differences in them. So the tricky part is that these spicules are even smaller. They're about 30 microns in diameter, and they're only about 1 millimeter long. They're kind of obnoxious to handle, but we did the exact same experiments on the Tethya aurantia spicules, and those are shown in red triangles here compared to the yellow circles for the euplectula spicules. So red is no layers, yellow is with layers. So that you can see for larger A over D, we can't really see a difference between the two. For very small A over D, we can see that the euplectula spicules do have a significantly higher initiation toughness. And same thing, getting work of fracture for the monolithic spicules was actually quite a bit harder because when we're computing work of fracture, we need to have a continuous force displacement response indicating that there's no unsteady crack growth and unaccounted for kinetic energy when we're doing that incremental energy balance of external work versus elastic energy and energy dissipated by fracture. So there's far fewer samples here. But we see that they do have a lower average R value as well. However, and this was the concluding slide I had shown before, when we compute the ratio of initiation toughness and average R of the architectured version of a material, so like a euplectula spicule or nacre, bone, antler, conch shell, to their constituent ceramic component. For euplectula, we used the Tethya spicule, and for nacre, bone, antler, and conch, we used references from hydroxyapatite and aragonite for those materials. You can see that this ratio really says how much the toughness is enhanced, both in a metric of initiation toughness and sort of total or average energy dissipated in this average value of R. And so we can see for the euplectula spicules, the enhancement both in initiation toughness and work of fracture is significantly less than these other materials. And that we had was why. Why is this material, which many people speculated also has this crazy toughness enhancement, why doesn't it achieve like these other materials? So this was work that I did in collaboration with my batchmate Kaushik Vijay Kumar, where he had developed a phase field model for fracture. So we looked at 2 representative cases. One has a planar interface and one has a curved cylindrical interface. And these so that the volume of the samples was the same. But the only thing that changes in these materials is not constitutive behavior, just fracture toughness. So we specified a interfacial fracture toughness GI, much less than the bulk material, in a very thin interface of a cylindrical shaped interface and a planar, this flat interface. And so by spatially varying fracture toughness with the same elastic properties, using a regularized variational fracture method or a phase field fracture method modeled after Blaise Bourdon's work, we looked at the response of these two model systems. So the planar system, you can see the crack growth shown in red as the damage field on the top, and on the bottom we have our force displacement response. So you can see a jump in the force displacement response, much like you'd see in Nacre or even the architectures that Dr. Yin just presented. And this jump corresponds to crack arrest at the interface, or a Cook-Gordon mechanism, if you're familiar with that, as well as it being accompanied by some deflection. But the deflection at the interface doesn't actually dissipate that much energy because the toughness of the interface is so much lower. If we look at the same thing for the cylindrical interface, what's very interesting is that we don't have that same jump in the force displacement curve. We still see the crack deflection at the interface, but we do not see crack arrest occurring. And we attribute this to the geometry of that interface. And that's work that's still being explored in the Kesari lab from, I think, a computational and a theoretical standpoint, looking at the role of interface curvature on both promoting or suppressing toughening mechanisms like the Cook-Gordon mechanism. And so if you want to learn more, the design of the testing system was shown in this, the first publication, and the most of the results that I showed today are in the second presentation. And in one of the questions that I had received in the chat by Professor Jairam, there is, we actually referenced their work a lot in the design of this experiment. So I wanted to sort of acknowledge that and say thanks as well.
Thank you, Max. I mean, typically with curvature, we expect an increase in stiffness, right? For example, but I mean, it's interesting that it could also affect toughness. I'm not familiar with how, but it's an interesting idea, right?
Yeah, I mean, in sort of brief, you can think of it like as the crack is growing towards the interface. If it's growing towards a flat interface, it sees a plane of weakness and can sort of nucleate daughter cracks ahead of the primary crack anywhere in that plane. Whereas if you have a curved interface, the crack's only really seeing a line of weakness. It's seeing that first cusp of the interface, and as you move further along the interface, away from that line, you're also moving further from the crack tip, so the stress is lower there.
So it's very constrained in how it can grow. Is that
exactly? It's constrained in how it can nucleate daughter cracks, and this nucleation of daughter cracks and that as a mechanism for redistributing stress is the primary way that toughness is enhanced through this like Cook-Gordon-like mechanism, or cracks nucleating at weak points ahead of the crack tip. And so that's less able to happen if you don't provide a very large surface for which it could happen. If you flip the curvature and looked at instead of a convex, a concave curved interface, it could actually promote that type of mechanism and potentially enhance toughness, but we didn't really investigate that.
There are people waiting.
Hi, Dr. Monn. This is Jen. Yeah. So like, yeah, because I'm quite interested in the the studies on the spongy spicules. But I was wondering, like, I know that those layered structures in general, like if it's continuous layer structure, they usually don't perform as well as discontinuous structure in terms of toughness. But I was wondering, like because I know that the spongy spicules, they have like layered structures, like concentric rings. But the thing is, like along the axis, is it like always continuous or also
it
like
is, yeah.
second?
No, no, it's continuous along the axis. Yes, yes, and I totally agree that segmentation can play a huge role in redistribution of stress, as you had shown, and involving a greater volume in that energy dissipation, which is partly, I mean, it's again a strong motivator for really thinking critically about the initial claims that this architecture produces toughness enhancements like nacre. They're often cited together. They're often talked about together, but I think through a combination of the experiments that you showed and the experiments that I showed, I think it's clear that we need to be a little bit more careful when we're sort of talking about these materials at large and really start differentiating with a higher level of granularity about the advantages of certain architectures over others.
Yeah, I think, I'm not sure if you are familiar with some like Corning. They did some very highly flexible glass, like brittle glass. It's very thin glass, but you can bend it quite a lot. But they're still brittle. Actually, I received some samples and they're still very brittle.
Yes.
Maybe is it possible that the mechanism between brittle glass and also sponge spicules has some similarities? Because I know that the mechanism of brittle glass is that you kind of remove all the you know, the edge defects and all those things, and so that because glass itself, I mean, if there's no defects, glass is a fascinating materials, and the brittleness and also all those like the low damage tolerance coming from the defects when you do the manufacturing and all those edge defects. So I was wondering, because the sponge spicules are in a very small scales, so it is possible because the absence of the nano and micro defects within the each layers, causing the highly the flexibility of the high flexibility of the spongy spicules.
Yeah, yes. So I mean, I think to address the 2 points there, the first is about this idea of materials becoming less sensitive to flaws at the micro or nano scale. I think that we can show that that is not the case by comparing, again, the euplectula spicules to the monolithic ones, the Tethya spicules. We don't really see that same kind of size effect as you'd expect out of work from like a professor like Huajian Gao, that type of thing. But I do think that your point about suppression of defects is very important. And I agree. I think the outer layers of a spicule are likely sacrificial, that if they take most of the damage and most of the interior layers are defect-free or near defect-free, that can greatly increase the strength of the spicules. I also have done some other work at looking at other internal deformation mechanisms that could allow greater flexibility in the spicules. So this is, if you can see the slide I have up, this idea of if you took like a ream of paper and stapled it on all the sides, it would be stiffer and it would fail sooner, like tear sooner than if you let all of the layers slide past each other.
Okay. Yeah, and also, is it possible that it is associated with how the micro defects and nano defects align, like orientated, like for example,
100%.
Yeah.
100%. Yes. Okay. But that's something that hasn't yet been quantified. It's very, very hard to measure at this scale for sure.
Okay. I see. Yeah. Thank you.
Of course. Thank you. I see a few other questions in the Q&A.
Right.
So in terms of the ceramic being broken into 2 pieces, what's the source of that energy? A number of toughening mechanisms, like Dr. Yin has showed in his presentation, that you can have large-scale tablet sliding, you can have dissipation at the interfaces due to viscoelasticity of the protein in the interfaces. You can have crack bridging. There are a very large number of mechanisms that can precipitate these type of enhancements. And so we were mostly focusing in this work on just one, being that Cook-Gordon effect, or nucleation of cracks at weak interfaces ahead of the primary crack tip. And the next question about higher toughness for short cracks. Yeah, the higher toughness for short cracks, that's something that we're still honestly looking into. But our first take at it is really, it's this one, is really that as you move towards the periphery of the spicules, the cylindrical layers get thinner and thinner and thinner. And so the effect of these type of mechanisms where you have daughter crack growth, crack arrest will become more and more significant for short cracks because they're going to be able to see more interfaces in front of the crack tip at the same distance. If that makes sense.
Yeah, if I may just continue with that question. The reason I asked this is even in monolithic materials, we are seeing that for very cracks, there is sort of a spike in the fracture toughness compared to the rest of, the longer.
Yes.
So I don't know. I was not sure whether it's the effect of the architecture in the material or whether it's something to do with short cracks versus long cracks.
So I think in this case, it is effect of the architecture. You can see that in this plot on the left, where we have similar crack lengths in terms of A by D for the monolithic sort of reference, the TA, Tethia aurantia, and you don't see that same increase. But I agree with you. I'm also aware of such studies looking at monolithic materials with very short cracks. But I think that primarily that has to do with, or my understanding is primarily that has to do with process zone sort of, the process zone not being a steady state. This was some work by Professor Subra Suresh a while ago. But yeah, in this case, I do think it is architecture related.
Sure, yeah. I had another question about the experimental method itself. When you make these kinds of notches, since we also worked on clamped conditions, so we always found that the notch was difficult to maintain it sort of straight. So when you make an assumption that it has a straight cross section in the circular, I mean to say the straight front of the notch in a certain section, that I guess is an assumption, right? So
we imaged them after they failed. So you can see a couple micrographs here where, again, the green is the macro notch and the orange is the micro notch, and you can actually see the notch root. And yes, maintaining straightness was very tricky, and that was part of the reason we cut the notch in 2 steps, and it was also part of the reason we had a lot of trouble with getting down the process conditions for the ion cutting in order to maintain the straightness of that notch.
This is great. Thank you very much.
Yes, of course.
Max, we are close to starting the next session. Thanks very much for taking time to answer, to finish your presentation and answer these questions. I hope that you'll stay back and listen to Dr. Colvin also. In about 5 minutes time, we will start the next session.
Thank you very much. Thanks again.
Thanks, Max.